Hello. The title of my presentation is Potential Risks of Dengue Fever Outbreaks in Japan and Laboratory Diagnostic Testing. As dengue fever is the most problematic mosquito-mediated viral infections in the world, in August 2014, in and around Yoyogi Park, there was an outbreak of dengue fever. It's been 6 years since then, but in fact, there were cases of dengue fever here in Japan last year. Dengue fever is still prevalent in tropical and subtropical regions. I have no conflict of interest to disclose. First of all, let's look at the blood feeding behavior of Aedes aegypti. In Japan, dengue virus is carried by Aedes albopictus, but a major vector of dengue virus is Aedes aegypti. And you're looking at the blood feeding behavior of Aedes aegypti. What you consider as a needle is called proboscis. In the teeth, there's a long, slender stylet. Both Albopictus and Aegypti belong to genus Aedes. Aegypti is known as a carrier of yellow fever virus, but Albopictus also mediates yellow fever. Zika fever and chikungunya fever were also mediated by Albopictus and Aegypti. It is reported that there is a transmission of dengue virus between monkey and mosquito, but generally the major infection route is between humans and mosquitoes, with the human being an amplifier for the sustained outbreak. Now, as you can see, the abdomen is bulging in red and the proboscis is pulled out. Japanese people tend to believe that mosquitoes are active during night. However, Albopictus is active during daytime. You can get bitten easily in early morning and also in the afternoon towards sunset. When the sun is too strong, Albopictus hide behind trees and behind leaves, so it is an ambush type of mosquito. Culex tridentyrhynchus is a vector of Japanese encephalitis. This mosquito is active in the evening but becomes less active in the midnight. But Culex pipiens continue to be active even during midnight. So when you're sleeping, sometimes you hear mosquitoes, and that's Culex pipiens. This type of mosquito does not carry dengue virus. The northern limit of habit for Albopictus is moving northward. Up until 1950s, the northern limit was south of Fukushima, but in year 2000, it went further north to the south of Iwate, and in 2016, a settlement in Aomori was confirmed. Albopictus requires an average temperature throughout the year of over 11 degrees Celsius. In 2019, 457 dengue fever imported cases and 4 domestic dengue fever cases were identified. Last year, in 2019, there was an outbreak of chikungunya fever in Myanmar and Thailand, and there were 49 chikungunya fever cases imported in Japan. We had 461 dengue fever and 49 chikungunya fever, and 11 of the chikungunya fever were imported from Myanmar to IG. When we have more people traveling back and forth, there could be an increase of the dengue fever imported cases. So the COVID-19 situation helps reduce the risk of dengue fever outbreak here in Japan, but we will not be closing our doors forever. This shows the weekly number of dengue fever patients reported in 2019. You can see the risk is the highest during the Olympic Games. The Games were postponed, but they take place in the same year, so if you look at the time lag of latent period and actual diagnosis and report, the actual infection may take place a week or two before the Games. We do not know how many people will come from overseas during the Olympic Games at this point, but it is important that we assume a certain level of risk. Dengue virus belong to family Flaviviridae, and they are similar to Japanese encephalitis virus, West Nile virus, yellow fever virus, and tick-borne encephalitis virus. There are 4 serum types, types 1 to 4. The gene sequence of these 4 have a homology of between 60 to 80%, so it's not too close. However, they exhibit the same pathophysiology of dengue fever. These viruses can often show cross-reaction, but the cross-defense level is not high. The serum types can be identified by comparing the neutralizing antibody titer, but you can use PCR too. So there's a confusion with genotype, but we are working on serum type. Of course, you can use the serum types to further identify genotypes. This is the first infection of pediatric dengue virus and the frequency of dengue hemorrhagic fever and shock syndrome for reinfection. Once you are infected by type 1, you would have defense immunity for a sustained period, but for other types, although there's cross defense to some extent, this will decline and you would become susceptible to other types of dengue virus. At the time of the second infection, sometimes the disease gets more severe, leading to dengue hemorrhagic fever frequency to increase to 2%. Dengue shock syndrome frequency for first infection is 0.007%, but for reinfection, the frequency goes up to 1.14%. For third and fourth infection, more people will have more antibodies that serve as cross-defense. Among the dengue fevers patients, viremia occurs in acute phase. Using real-time RT-PCR, genes can be detected during the high fever period. With the antibody increase, the PCR sensitivity comes down, but NS1 antigen can be detected a few days to a week even after the body temperature is lowered. So NS1 antigen testing can fill the gap between PCR and IgM testing for a better laboratory diagnostic testing. IgM testing with capture ELISA can detect the positivity 4-5 days after onset, and NSI antigen can be detected after a week since the body temperature is lowered. The gene detection rate of dengue virus from blood sample is not 70% but very high, but this level comes down when the fever is subsided. Unlike airway samples of COVID-19, blood sample is homogeneous. Whether you take samples from your right arm or left arm, the amount of virus is the same. Dengue virus can also be detected from saliva and urine, but this is not applicable to 100% cases. When you detect a virus, you can see the detection for a few days even after the end of viremia. This is the proliferation process of a virus. You have the processes like attachment, penetration, uncoating, and then gene expression and translation take place. Protein is synthesized, viral particles are formed, which are then released outside of the cell. Gene expression and translation may take place within cytoplasm or nucleus. In case of dengue virus, they take place in cytoplasm. Once you are bitten by a mosquito, the dengue virus in the saliva will infect the Langerhans cell, which is a macrophage, and proliferate in the lymph node, and the proliferated virus will go into bloodstream. So just because you are bitten by an infected mosquito does not mean you would have your virus detected from your blood sample in just a few days. A dengue virus is a positive single-stranded RNA. So to proliferate the gene, you need to have a reverse transcription from RNA to DNA. RNA is easily degraded by enzyme like RNase and heat, so before the use of PCR it has to be converted to DNA. When we talk about RT-PCR, RT stands for reverse transcription, not real time. RNA virus gene detection is often being studied with real-time RT-PCR. In summer 2014, there was a dengue fever outbreak caused by type 1 of virus, which was also seen in Singapore and Malaysia. The mosquito cannot survive in temperate winter, so this outbreak ended in November in 2014. And starting from 2015, local governments tried to eradicate the mosquitoes, larvae and adult during summer, so there was no case of dengue fever reported. But last year, there were 4 cases of dengue fever in Japan. I will talk about this in more detail later. This newspaper article says that the students traveled to Kyoto, not that they were on our overseas trips. A dengue outbreak in 2014 was caused by type 1, and all gene sequences were identical. So the infected patient from overseas was bitten by mosquitoes. It's not perhaps single albopicus, but there were several mosquitoes biting the same patient, and after a week or 10 days the mosquitoes become infected, and those mosquitoes blood fed the visitors of the park and those who live in the park, and this is how there was an outbreak in a multiplying effect manner. When most of the park was closed, those who live in the park moved to Shinjuku Central Park, where there were several cases of dengue fever reported. In 2019, there are 3 cases of dengue fever infection among those students who had a school trip. They belong to the same school. Patient A is a male in his teens. Between 18th to 20th of September, he traveled to Nara and Kyoto, and he had a fever on the 27th, and he visited a clinic. He had a fever, headache, joint and bone pain, leukocytopenia and thrombocytopenia, and he was admitted to a hospital and diagnosed as dengue fever. And patient B is a woman in her teens. Again, upon her return from school excursion trip, she had a fever on the 26th of September. She had fever, rash, leukocytopenia, thrombocytopenia, and she was admitted to a hospital. Type 2 dengue virus was detected from this patient. A third case did not show gene detection but had an episode of mosquito bite, and with ELISA, dengue virus specific IgM antibody was positive. Dengue virus specific IgG antibody was also positive, and there was a significant increase of neutralizing antibody titer for dengue virus type 2. And this is a suspected case in Okinawa. On the 30th of August, 4 days after the return from Nepal, the patient had a fever, and from the 3rd of September, the patient had rash and diarrhea, and dengue fever was suspected. The patient was NS1 antigen positive, IgM positive, and IgG positive, and diagnosed as dengue fever. Because of the serological diagnosis being confirmed, genetic test was not performed. The patient traveled to Nepal in between 16 to 26 of August, so this was an imported case. And with regards to the second infection cases, this is patient X. She's in her 70s. In August 16 to 26 in 2019, she visited Nepal together with the previous patient who is a family member. 20 days upon her return, on the 15th of September, she had a fever. On the 17th, she was clinically diagnosed as suspected dengue fever, and she had a test on the 18th, and because she was NS1 antigen positive, she was diagnosed as dengue fever. She had diarrhea and abdominal pain and vomiting of blood. She was admitted to a hospital on the 19th, and she had a gene testing, and it was identified as the dengue virus 2. The lowest platelet count went down to 20,000 per microliter. She also had hypoproteinemia, and she developed dengue hemorrhagic fever, and she was discharged on the 30th because she traveled to Nepal, because she lived with the family member who was the first patient, and because her living environment was such that there were many mosquitoes and were easily got bitten. We consider that it is possible that this case is the secondary infection case that took place around her living environment. A year before the outbreak of dengue fever in Japan in 2014, there was the following case reported from Germany. In 2013, with the direct flight from Frankfurt, the patient arrived in Narita. The traveler went to Ueda, Fuki, Hiroshima, Kyoto, and Tokyo, and the patient left Narita to Frankfurt with a direct flight on the 31st of August. The patient actually had a fever on the 3rd of September, and she had nausea and erythematous papular rash, and she was admitted to a hospital on the 9th of September. On day 7, with the PCR, she was genetically negative, but she was IgM positive and NS1 antigen positive, and she was confirmed as a dengue fever patient. The latent period of dengue fever is between 2 to 14 days, but usually it is between 4 to 7 days. So it's possible that she got infected either in Kyoto or in Tokyo. At the time of our outbreak in 2014, our German colleague interviewed the patient, and we learned that she actually visited Shinjuku Park while she was in Japan. The German traveler visited popular sightseeing spots in Japan. When we have more inbound travelers from overseas, we have to be careful about these spots, and we shouldn't take light of the case that was seen in 2013, as this is a key for good infectious control. Between 1942 to 45, there was the largest dengue fever outbreak in the temperate zone in Japan. This shows the number of patients per city and per year. In 1942, in August, dengue fever was reported in Nagasaki that spread to Sasebo, Osaka, and Kobe. The outbreak started in summer and subsided in winter every year, and the outbreak spread to Fukuoka, Hiroshima, and Kure. In 1944, there were 100,000 patients in Osaka and Kobe. This was during the Pacific War. So the soldiers from overseas came back to Japan with infection that started the epidemic. And because of the military expenses, there was not much budget to control the mosquitoes. But with the end of the war, there was a dramatic decline in the dengue import from overseas, with no epidemic that followed. Dengue fever is not a new infection at all. In Okinawa, even before war, there was an outbreak. This paper was written by Dr. Koba in 1932. There was a dengue fever epidemic in Taiwan. The risk of dengue fever depends on the frequency of people traveling. This is from an older medical journal, and this paper was published in April 1920. This paper is titled Study of Dengue Fever. The blood from dengue fever patient was tried on different animals like mouse, rat, guinea pig, rabbit, dog, goat, dove, and lizard. But dengue fever-like symptoms were not observed. It was a Japanese scientist who isolated the first dengue virus in the world. In 1938, dengue virus type 1 was isolated from a patient in Nagasaki, and based on the name of the patient, this is called dengue virus type 1 Mochizuki strain, and was recognized as the first isolated strain of dengue virus. the hard-working effort of the Japanese scientists led to the first isolation of dengue fever virus. This is my last slide. This is about how we should be prepared for tropical viral infection. With the global warming and increase of the temperature in Japan, we will see more of the Egypti in Japan, and they lay eggs in small water pools. So there are many possible areas where these mosquitoes can settle. It is also important to promote multiplex system for gene testing for mosquito-borne virus. We should assume viral mutation, so it will be useful to have a database of a gene sequence, but virulence cannot be decided just by sequences, so viral isolation would be important.